The humoral response is the body's way of combating antigens and other foreign substances. Pathogens first enter the body. This can be through transmission in the air, through ingestion of food, or retrieved from contaminated surfaces. Once the pathogen enters the body, it begins to interact with the macrophage. The pathogen has many proteins on its surface called antigens, while the macrophage has a series of class II MHC proteins on its surface. Additionally, it contains many lysosomes that hold digestive enzymes and others that hold class II MHC proteins. This is the start of the activation phase. When the macrophage gets in contact with the pathogen, it engulfs it in a process called phagocytosis. The resulting product is called a phagosome. The phagosome then fuses with a lysosome that contains enzymes capable of breaking down its contents into antigen fragments. Within the cell, the processed antigens then combine with class II MHC proteins. This complex is then transferred to the surface of the macrophage in a phenomenon called antigen presentation. These macrophages are now considered antigen-presenting cells. Another group of cells, called T helper cells, then come and attach to the antigen complexes with their receptors. This causes a response in the macrophage to produce cytokines called interleukin-1 and sends them to the T helper cells to activate it. The T helper cells then produces its own cytokines, which causes it to mass replicate into more T cells with the same antigen-specific receptors. The next step in the humoral response is the effector phase. Within the immune system, there are cells called B cells, which contain IgN receptors, lysosomes, and class II MHC proteins. The B cell in this case has IgM receptors that are specific for the same antigen from the pathogen engulfed by the macrophage originally. When an antigen makes contact with the IgM receptor, it is ingested in a process called receptor-mediated endocytosis. Similarly, as before, the antigen then combines with the lysosome and is broken down into antigen fragments. It is then combined with the class II MHC protein. This complex is also transported to the surface of the cell membrane. Then, a T helper cell from the clone of cells from the activation phase come and attach to the presented antigen displayed on the B cell. The T helper cell then releases cytokines to the B cell, which stimulate it to proliferate into many identical cells. The B cells then develop into antibody-producing plasma cells, or into memory B cells. The memory B cell is sent to the lymphatic system, which will allow the body to quickly identify and remove the antigen if it makes its presence in the body again. These plasma cells have a lot of endoplasmic reticulum, which allows it to produce large amount of antibodies specific to the original antigen. These antibodies then bind to the antigens in a process called agglutination. Agglutination forms long chains of antigens and prevents them from causing any damage. After, macrophages then come and destroy the agglutinated complex, and the body is rid of the antigen. The humoral response is now complete.